Hi class, in this recording we're going to focus on the small intestine. Let's look at the small intestine. The big thing we need to emphasize is that we have chemical digestion and nutrient absorption in the small intestine. Those are the two biggies of the small intestine. And other take-homes of the small intestine is that it has the most surface area of any part of the alimentary canal. It is the single longest part of the digestive tract, approximately 5 meters long in a living person, and then post-mortem in a cadaver it's 8 meters long because of all the longitudinal smooth muscle within it relaxing. We call it the small intestine because it has a narrow diameter, not because of its length. The diameter of the small intestine is approximately 1 inch. So it's quite narrow relative to the colon or some other organs within our alimentary canal. As we look at the small intestine, it's going to coil upon itself over and over and over again. And it's going to make up the main segment of our abdominal pelvic cavity. It's just inferior to the stomach and liver, and it's going to be divided up into 3 regions. The duodenum is the first region, the jejunum is the middle, and then the ilium is the third and final region. As we look at our small intestine, the small intestine begins at the pyloric valve, which goes over the opening known as the pylorus. That's a common sticking point, particularly on lab exams. The opening is the pylorus. The valve is the pyloric valve. And the duodenum, or duodenum, it's kind of a potato-potato situation as far as I'm concerned. The duodenum is begins immediately after the pyloric valve and is quite short, approximately 10 inches long. So I'm going to shift to blue here. The duodenum is, I'll go a little wider here. The duodenum is right here. Let's go really wide. There we go. So our duodenum is right here and extends 10 inches to the jejunum. And the jejunum is then that purple region right down there. So as we look at the duodenum, it's going to receive a lot of stomach acid in the form of the chyme draining out of the stomach. It's also going to receive pancreatic secretions from the pancreatic ducts and is also going to receive bile secretions from the common bile duct. Let's look at the duodenum. It arches around posterior to the transverse colon and ends at a flexion right here, referred to as the duodenojejunal flexure. Now, as we look at the duodenum, notably, since it's posterior to the transverse colon, that duodenum is going to be retroperitoneal. Most of it is at least going to be retroperitoneal. It's going to receive the chyme from the stomach, the pancreatic juice from the pancreas, and the bile from the liver and gallbladder. The pancreatic juice in particular contains a lot of sodium bicarbonate, which is going to neutralize the stomach acid that is being drained in through the pyloric valve. The fats are also going to be physically broken up, and to physically break them up and dissolve them in water, we have emulsification. And to say that emulsification is the same thing as dissolving is a bit of a misnomer. But it's the best single word synonym I have for emulsification. If you want a good example of emulsification, wash some greasy pots and pans and just start out with no water, or no dish soap. And then after a little bit of trying to wash your greasy pots and pans with no dish soap, add a couple drops of dish soap and watch the big difference that happens when the dish soap emulsifies the fats. We also find the pepsin that comes from the stomach is going to be deactivated in the duodenum because the duodenum has this alkaline pH, whereas the stomach has an acidic pH. The pancreatic enzymes that are secreted from the pancreatic juice are going to perform a myriad of chemical digestion functions. There's a lot of them. As we look at the jejunum, and this figure is purple, and it's about 1/2, just a skosh less than 1/2 of the small intestine, and it's going to immediately follow the duodenum. So we go from the duodenum and flow into the jejunum. It's about 1 to 1.7 meters in a living healthy person, and it's going to have closely spaced circular folds that go back and forth. Also notable is that it has a relatively thick and muscular wall and has a very rich blood supply, which gives it its bright red color. So when you're looking at the abdominal pelvic models of plastic torsos in lab, you'll note that the jejunum of the model is going to have that bright red color or slightly darker coloration relative to the ilium, which is the third and final portion of the small intestine. As we look at, let's connect the color to this next bullet point here. So it has bright red color because of lots of blood supply. That blood supply is important because we have nutrient absorption. And as we absorb carbohydrates, proteins, and nucleic acids, we are going to dump those into the blood capillaries or the bloodstream. So that part of the small intestine, the jejunum, that has a lot of blood, is also going to have lots of nutrient absorption. However, after the chyme has gone through the jejunum and has arrived at the ilium, there's not as many nutrients left, so we don't have as much nutrient absorption later on when we get to the ilium. As we look at the ilium is approximately 60% of the small intestine post-duodenum. And as we look at this ilium, it's about 1.6 to 2.7 meters in diameter, depending on the individual. Because it is not going to have as many, the chyme within the ilium, it doesn't have as many nutrients. The ilium doesn't need as many blood vessels. So the ilium is less vascular, has less vasculature, and it'll be paler or pink in color relative to the jejunum. It's also worth emphasizing that the ilium is where we start to have increases in bacterial concentration. So we're going to have lots of lymphatic nodules associated with the ilium. And these nodules are clustered opposite of the mesenteric attachment point. As we look at the ilium with the naked eye, these lymphoid nodules become larger and larger as they get closer and closer to the large intestine, where we have feces and a ton of bacteria. As we go from the ilium to the large intestine, we have the ileocecal junction. And this is not a, this ileocecal junction, where the small intestine ends, has a very important structure, the ileocecal valve. And this is a valve, not a sphincter. I know it says sphincter right here, but this is more of a valve in that it has a flap of tissue that allows for one-way passage, typically speaking. So we have a flap of tissue combined with the sphincter muscle so that we can have chyme entering the large intestine from the ilium, but we have a very minimal amount of feces going backwards into the small intestine. This ileocecal valve is going to protrude from the ilium into the cecum of the large intestine, the main blind pocket, that first part of the large intestine. As we look at the jejunum and ilium, these are both going to be intraperitoneal. They are part of the abdominal pelvic cavity and are going to be covered with the peritoneal serosa. Now, in the small intestine, to aid with our nutrient absorption and digestion, we need to have a very special kind of epithelium. We have a simple columnar epithelium, but as we look at the simple columnar epithelium, it has a lot of villi within it, and then as we look at those villi, the individual villi of the simple columnar epithelium are coated with microvilli. So between the villi, the microvilli, and all the circular folds, sometimes referred to as the plicae circulares, we find that the small intestine has a very large surface area. The musculatura externa within the small intestine has a thick inner circular layer and a much thinner outer longitudinal layer. That thick inner circular layer helps to form the circular folds, which increase surface area. Compared to the large intestine, the small intestine, even though it's narrower in diameter, has a much larger internal surface area. And it's because, again, of those circular folds, the villi, and then the microvilli. So as we look at a villi, you can think of a villi as an extension or a little bump that has a lot of individual little cells on it. And then each individual little cell has a bunch of microvilli forming a hairy brush border enzyme. And it's those microvilli that really increase the surface area more than anything else. And this is a figure that, or a factoid that's always stuck with me since I was in high school. The surface area of the small intestine is greater than the surface area of a standard tennis court. So when you're looking at an adult's small intestine, it has an enormous surface area because of the circular folds, the villi, and the microvilli, all maximizing surface area so it can maximize the rate of nutrient absorption. Now, as we look at those villi within the small intestine, those villi are extensions made of individual cells, and the individual cells of the villi are each going to have microvilli on them. The villi themselves give the mucosa a fuzzy-looking appearance because they are still quite small. Notice how you can see individual cells within that villi figure. There are two kinds of cells. We have the enterocytes. Enterocyte is kind of a generic term that refers to intestinal cell. A term I prefer more is absorptive cell. It's a descriptive term. It tells what is it's a descriptive term based on the function as opposed to the location of the cell. And then, of course, we have the classic goblet cells, which make lots of mucus. The epithelium of the small intestine is going to have a bunch of tight junctions to keep digestive enzymes in the small intestine. And as we look at the villus in the core, let me clear the screen here. In the center of the villus, we have a blood capillary and a lacteal. The blood capillary is going to aid with the absorption of carbohydrates, proteins, and nucleic acids, whereas the lacteal within the center is going to be used for the absorption of lipids into the lymphatic system. Now, as we look at the microvilli themselves, those small extensions of the cell membrane of the individual cells that make up the villi have a fuzzy brush border appearance. And on the apical surface of each cell, the microvilli are approximately 1 micron high, so they're quite small. You're going to need a very high-powered microscope, a good microscope to see that. And if you're able to zoom in even more, you would need an electron microscope at this stage, you could see that there are digestive enzymes embedded within the plasma membrane of the microvilli. So in gray here we have a plasma membrane, and then we have digestive enzymes. And the chyme will just flow downstream, and as the chyme flows downstream, molecules within the chyme, food molecules, rub up against the enzymes along the border of the intestinal wall and are chemically digested. Oh, 1 second, I have to sneeze. There we go. So that chyme needs to brush against the brush border enzymes for chemical digestion to occur within the small intestine. We also are going to have some glands specific to the duodenum. The duodenum, again, is that first part of the small intestine, and within the submucosa of the duodenum, we're going to have a bunch of bicarbonate-rich mucus. And it's important that we have bicarbonates. Bicarbonates HCO3 with a -1 charge, and that HCO3 with one negative charge. Sorry about that. With that HCO3 with a 1 negative charge, it's going to be a basic ion. And that bicarbonate neutralizes the stomach acid, the hydrochloric acid that's being constantly dumped into the duodenum from the pyloric sphincter. We also find that we have a large concentration of lymphocytes within the laminopropycha and submucosa of the small intestine. And these lymphocytes in particular are going to function to intercept pathogens. They're going to be concentrated mostly within the lymphatic nodules at the very end, the ilium, right next to the feces of the small intestine. These lymphatic nodules were formerly referred to as Prior patches or Prior's patches. I'm a little older and crustier, so that's what I had to learn back in the day. Lymphatic nodules, though, is a more descriptive term. It's easier to comprehend. If you have any questions or comments about the small intestine, please feel free to post them on the class discussion boards or shoot me an email. And as always, class, happy studies.